Hello everyone. Our today's lecture is regarding the chapter of blood biochemistry. So basically we'll discuss all the biochemistry processes that takes place in the blood. And this today is the first class of this chapter and we have more to come in the future. So mainly today we'll be focusing on the proteins that are found in the blood plasma, also known as the plasma proteins. Now, plasma proteins, in order to understand more, we first need to understand some features or characteristics of the blood here. I think we all know that the total blood volume is around 4.5 to 5 liter in an adult. Now, if you allow the blood to react with an anticoagulant, that is any drugs that will prevent the coagulation of the blood, and then if you centrifugate the blood, it will allow the cells of the blood to be separated. By cell, I refer to RBCs and WBCs. And the supernatant that we will get from these reactions are known as plasma. Majority of the blood is made up of plasma, around 60%. The rest 40% will be made up of the blood cells that we have. Now, if you withdraw blood without anticoagulant and allow it to clot on its own, after about 2 hours, you'll get a liquid portion from that, which will be known as serum. So please make no mistake or confusion between the term plasma and the serum. So serum will be the liquid part after the clotting of the blood. And by after the clotting, I am referring to when clotting is completed, so it won't have any more the factors that are responsible for clotting, such as prothrombin and fibrinogen. Okay, so that liquid part will be known as serum, whereas the supernatant part that will be allowed to centrifuge after anticoagulation will be known as plasma. If you see the following images, maybe it will make you understand in a more clear manner. See here. So if you allow or withdraw blood and then allow any anticoagulant, it will prevent the coagulation of the blood. And then if you centrifuge it, you'll get something like this, where the blood cells that you have on the blood will deposit at the bottom, whereas the top portion, the supernatant, will be the liquid portion of the blood. The majority of the bloods, as we have seen, are plasma, around 60%. Plasma remains in the top, being the less dense one. The most dense one are the RBCs, so they'll settle at the most bottom part. And then we'll have WBCs and platelets, and then at the very top we'll have the plasma. See here the difference between the plasma and the serum. Plasma is the liquid portion of the blood, which does not have the blood cells, and that has been treated with anticoagulants. Then what is serum? Serum is a liquid part of the blood which have been treated with anticoagulants. By treated with anticoagulants, by that I am saying is, so it which hasn't been treated with anticoagulants, I want to say. So they have been allowed to clot, and after clotting, the liquid portion that remained is defined as serum. So basically serum will lack those coagulant factors such as plasma and fibrinogen, prothrombin. These factors will be lacking in the serum. Components of the plasma. The blood plasma is mainly majorly composed of water, 90%. It has plasma proteins and electrolytes. See here the plasma proteins is making up 6 to 8% of the total plasma. Other than these three major components, it also contains nutrients, hormones, blood gases, and waste as well. So let's define what is plasma protein. Basically, plasma proteins are proteins that are found in the plasma. They'll be found in a dissolved manner, and there will be a large number of plasma proteins mainly. They will include simple protein, conjugated protein, and they will perform a number of functions in the plasma as well. Now let's check out some important terms such as what is meant by total protein. Total protein of a normal plasma is around 6 to 8 gram per 100 milliliter. We know that the plasma protein that we are discussing here today are mainly consist of proteins such as albumins, globulins, fibrinogen. The albumin to globulin ratio is kept usually in a normal condition around 1:2:1 or 1.5:1. Almost all plasma proteins, we know, that are synthesized in the liver except for the immunoglobulins. And we have also learned that most plasma proteins are glycoproteins. The term total protein is very widely used in a clinical setup, mostly in order to diagnose liver conditions, how well our liver is functioning, etc. etc. See here, so it's one of the most common measurements in clinical lab. It includes mainly albumin as well as some globulin acids. The amount of total protein can alter based on the body's condition. For example, if we have changes in the balance of fluid, so if there is loss of fluid by dehydration, by excessive sweating, diarrhea, etc., the amount of total protein will basically increase. Conversely, if we dilute the blood by, let's say, through fluid therapy, or in cases of pregnancies, in cases of salt retention, the total protein amount usually will decrease. So if you dilute the blood, the amount of total protein decreases. If you lose fluid from the body, your total protein content of the blood increases. Now, these plasma proteins that we are discussing here today can be separated by a very easy mechanism in the lab, known as the electrophoresis and ultracentrifugation. Most widely, electrophoresis will allow us to separate this plasma protein based on the charges in the protein. So, based on the charges that we have, okay, we can very easily allow these proteins to be separated. The positively charged proteins will move towards the cation or a cathode, whereas the negatively charged proteins moves towards the anode. In this way, we can very easily separate the plasma proteins and know in detail about their amount, their content in the blood, which is very important for clinical setup. Now, these are the main plasma proteins that we find in the bodies basically. We have albumin that makes up 60% of the total proteins of the plasma. They are originated from liver. They help to maintain blood osmotic pressure. We have globulin that makes up around 36%. That comes under some subtypes: alpha globulins, beta globulins, and gamma globulins. Except for gamma globulins, which is immunoglobulin, the rest of the tools are made in the liver. They mainly helps with the transportation, whereas the immunoglobulin gamma made in the lymphatic tissue and plays roles as an antibody. The other major protein that is found is fibrinogen that makes up 4% of the total plasma protein, again synthesized in the liver, plays roles in blood coagulation. Now let's go into details about each of these plasma proteins. Now, albumin is the most abundant plasma proteins that we have, okay, in the blood plasma. It's a single polypeptide chain made up of 585 amino acids with 17 disulfide bonds. They are synthesized from a pre-proalbumin into a single peptide and then ultimately into our albumin here. The major portion of the albumins are produced by the liver. So to be precise, 12 grams of albumins are produced by the liver per day. Half-life of the albumin includes 20 days. Okay, as majority of them are produced by the liver, that's why in order to assess the liver function, we most commonly determine or check or measure the serum albumin, because this gives us an idea about or indication about how well our liver is functioning. The structure of the albumin, as you can see in the image, is somewhat called ellipsoid. It has a charge and also has three major domains in the structure. What are the main functions of the albumin? Number one, and the most important function, is that it helps us to maintain Osmotic pressure. Colloid osmotic pressures are pressure that are applied by the proteins in the blood plasma. So they have the ability to pull water into the circulatory system. So in cases where we have a reduced amount of plasma proteins, let's say we have loss of protein through our kidneys into the urine, the condition known as proteinuria, or let's say you are deprived, that is your diet does not consist of sufficient amount of protein. In these cases, there can be a reduction in the osmotic pressure, and this will leads to accumulation of fluid in the tissue spaces because we don't have sufficient protein to pull them in the circulation, so the fluid accumulates in the tissue spaces and may cause a condition called edema, accumulation of the fluid in the tissue spaces. See here, in normal conditions over here, water will be pulled into the circulation by the albumin. So it is not allowing water to remain in the tissue spaces. But when we have a low amount of albumin, water can accumulate in these spaces, and you can see it can cause swelling, okay, causing edema. The second function of the albumin is in transport. Albumin can bind to several molecules and help in transportation, such as metal ions, fatty acids, bilirubin, bile acids, hormones. So it plays a very, very vital role for those material which are insoluble. One of the most common one, I am giving you an example, is bilirubin. Third function is that it has a buffering action. As it is having a higher concentration in the blood, albumin helps us to maintain a normal pH. So it has a buffering ability. It has 16 histidine residues. This basically will contribute to the buffering action. Number 4 is nutritional function. All tissues or cells can take up albumin by the process of pinocytosis. So if any tissue is requiring any protein, it can uptake this albumin and then later on break down into simpler amino acids. So it can be also considered as a mode of transportation for amino acids. Albumin is also used widely to treat liver diseases, hemorrhages, shock and burns as well for this purpose. Number 5 helps to maintain the blood viscosity, that is the stickiness of the blood. Number 6 provides stability to the blood. Number 7 helps with immune function. Now let's check out some clinical aspects in regards to albumin. As we have already told you that albumin can bind to several molecules. Okay, so it can also alter the effect of certain drugs. Let's take the example of sulfonamides. Sulfonamides can cause the release of unconjugated bilirubin, which is insoluble. Okay, and especially in infants, if this large amount of unconjugated bilirubin, if it remains in circulation, it can lead to deposition of these bilirubin in the brain, a condition known as kernicterus. In case of liver disease or starvation also, we know albumin can decrease and it may cause edema. It can cause a condition called hypoalbuminemia, okay, which may be found in malnutrition or cirrhosis of liver. It can be also used for therapeutic purposes, for treatment, especially for burn victims, for patients with have history of hemorrhages. For them also we can use it. So albumin has a very important compound in the body and helps in several, and it serves several purposes as well. The second plasma protein we have is called prealbumin. Please do remember it also has another name called transthyretin. Okay, so mainly what is it? It's basically rich of the amino acids TRP. If you know what is the TRP amino acids, you can guess it if you don't know. The 20 amino acids that we have learned, okay, you know that TRP stands for tryptophan. So basically prealbumin is mainly made up of tryptophan, okay, and it has negative charges, so this protein will migrate towards the anode compared to albumin which has positive charges, okay, and this also migrates faster compared to albumin in electrophoresis. It is considered a transport protein for thyroid hormones and vitamin A. Okay, and just like any plasma proteins, it can be separated by immunoelectrophoresis. The next plasma protein that we'll discuss is alpha 1 antitrypsin, also denoted as alpha 1 antiproteinase. In short, we call it alpha 1 AT or AAT. This is a plasma protein which is also a glycoprotein like others and made up of 394 amino acids. This basically is a globulin protein, mainly alpha globulin part, subtypes of the globulin. Normal concentration is around 76 to 189 milligram per deciliter and also found in tears, lymph, bile, semen and amniotic fluid as well. The clinical significance of alpha 1 antitrypsin is that it can be used to it can be referred with the condition called emphysema. When we have very low level of alpha 1 antitrypsin, this may cause distension of the lung and can cause collapse of the alveoli basically. So alpha 1 antitrypsin is very important to maintain the lungs structure specifically because it prevents the expansion of the lungs to an extent where the lungs can collapse. So in cases of lung infection or where we have increased activity of macrophages, okay, certain enzymes can be released called elastase and that can damage lung tissues and thereby may decrease or have a effect on the level of alpha 1 antitrypsin. The second clinical significance is it can cause problems with the liver. Sometimes these proteins, alpha 1 antitrypsins, can get mutated and can form polymers. This may lead to liver damages, mainly due to accumulation of collagen and may cause liver fibrosis and liver cirrhosis. The fourth importantly, as you can see here, alpha antitrypsin can increase in acute phase responses and by estrogens. For example, condition like in infection, it can greatly increase in number also. Secondarily, in respiratory distress, it can become low. In severe pancreatitis or in condition where you are losing proteins, the concentration may decline. See here, smoking can cause oxidation of methionine 358 to methionine sulfur oxide and thereby can inactivate this protein known as alpha 1 antitrypsin. The fourth one, important one of the plasma protein is RBP or retinol binding protein. It's a very small protein and mainly a transport protein for all transretinal. So mainly it will physiologically form vitamin A. Zinc will be required for this process and a decreased concentration of retinol binding protein can be seen in cases of liver diseases, protein malnutrition and in case of any acute phase reactant. Zinc deficiencies also can be characterized by low serum concentration of both vitamin A and retinol binding protein. The fifth plasma protein is known as haptoglobin. Haptoglobins are known to bind with free hemoglobin that are produced by the destruction of RBCs and thereby they are known to conserve the body's iron and preventing the renal filtration of toxic hemoglobin. Haptoglobin cannot pass through the glomeruli and thereby can prevent the loss of hemoglobin into urine. Haptoglobin can bind with several forms of hemoglobin. It can bind with adult hemoglobin, fetal, hemoglobin S, hemoglobin D, okay, but those which lacks the alpha chain of hemoglobin, to that the haptoglobin cannot bind. The normal protein concentration of the haptoglobin is around 3 gram to that of the hemoglobin, and the kidney can process up to 5 gram of hemoglobin per day. Low level of plasma concentration of haptoglobin can be known as hemolytic anemia as well. Clinical significance of haptoglobin. Haptoglobin concentration can increase by corticosteroid hormones. Okay, haptoglobins like ceruloplasmin are weak and also known as late reacting acute phase protein. Haptoglobin concentration may increase in nephrotic syndrome and in biliary obstruction, can decrease in disease such as hemolysis or hemolytic anemia, hemoglobinopathies. In cases of severe blood loss, haptoglobin may be totally decreased or depleted and may require up to 1 week of time to come back to normal level. The next one on the plasma protein is something known as ceruloplasmin or CER. It's a blue colored copper containing protein. It's a part of also the alpha 2 globulin, and you see it contains 6 to 8 copper atoms per molecule. So mainly what happens, they are synthesized by the hepatic cells, that is liver cells, and also a small portion can be synthesized by lymphocytes and macrophages. What is the importance here? The normal plasma half-life of ceruloplasmin is 4 to 5 days. Please keep in mind that ceruloplasmin is also called as peroxidase, an enzyme that helps in the incorporation of iron into transferrin. The ceruloplasmin is known to carry 90% of the copper tightly to it. Majority of the plasma ceruloplasmins are involved with copper binding, and the rest of other copper that are left around are bound by albumin and transcoupling. Low level of ceruloplasmin are associated with more free coppers, basically, and this may create a condition called Wilson disease, also known as hepatolenticular degeneration. It may cause malnutrition, nephrosis, as well as cirrhosis. Usually, the Wilson disease is an inherited disease where there is too much copper in the body. The too much copper can be damaging to our liver as well as to our nervous system. So if we have a low level of ceruloplasmin, it means that there will be more freely circulating copper. Usually this condition can be treated with a chelating agent. Chelating agent means something that can remove the copper from the body. This includes a drug called penicillamine. The first treatment that is used, this may chelate the copper and help the removal of copper from the body via urine. Wilson disease is mainly due to defects in the gene ATP7B. It's an autosomal recessive disorder. In these cases, the body is able to normally absorb copper but are unable to normally excrete copper into the bile. And as a result of these, copper will accumulate and ultimately will deposit into several tissues in structures like liver, brain, and it has a typical specific clinical manifestation called Kaiser-Fleischer rings, corneal rings you'll find in the eyes. It may cause hepatitis, cirrhosis, and as if it affects the brain, may cause behavioral changes, personality changes, tremors, and etc. Next plasma protein is called transferrin, or also known as siderophilin. Again, it's a glycoprotein, but of the subtypes of beta globulin. It has two iron binding sites in a single polypeptide chain. Their roles is to mainly bind with ferric iron. They can transport the iron in the plasma as ferric ions and protect the body against the toxic effect of free iron. Their half-life is around 7 to 10 days and they are used as an index for protein turnover. They are increased in conditions like iron deficiency anemia and in pregnancy, decrease in protein energy, malnutrition, cirrhosis, and nephrotic syndrome. Next one that are found in the plasma is acute phase reactant or acute phase proteins, APP or APR, both are right. These are basically a group of proteins whose plasma concentration will change in response to inflammatory reactions such as infection, surgery, trauma, myocardial infarction, malignancies, and so on. This includes C-reactive protein, alpha-1 acid glycoprotein, fibrinogen, haptoglobin, alpha-antitrypsin, albumin, and transferrin. It is very important for you to know which protein's level will increase in acute phase and which protein will not increase or decrease in acute phase protein. So this chart is very important and high yield. Please do remember. In acute phase reaction, the proteins such as C-reactive protein will increase mainly. See here, 1000-fold amount. Alpha-1 antitrypsin will increase, alpha-1 anti-chymotrypsin will increase, ceruloplasmin, fibrinogen, haptoglobin, these all will increase. Some proteins will decrease in level as well, such as albumin, transferrin, retinol binding protein. These will basically decrease. So please do remember which proteins will increase during acute phase reaction and which proteins will decrease. So first one is the C-reactive protein. This is a major content of the acute phase protein. So sometimes in order to evaluate any acute phase response, we usually measure the amount of CRP. As we have seen, CRPs will increase in a very high amount in cases of inflammation, acute inflammation, okay, and will ultimately reach to the peak around 48 hours. The normal level of CRP or C-reactive protein is less than 0.8 milligram per deciliter. Functions of the C-reactive protein is initiation of the process of opsonization and phagocytosis. So they play roles in defense mechanism basically. They will activate complement, activate neutrophils and monocyte as well as macrophage. So mainly they will be able to recognize any microbial organism and will initiate the host defense system, the C-reactive protein. The next one in acute phase protein comes is fibrinogen, also known as clotting factor 1. Their role is to mainly take part in blood coagulation, and they also act as a precursor for fibrin, a substance that is required for clotting. They are usually activated by thrombin. Usually fibrinogen will be converted into fibrin by cleavage of the arginine glycine peptide bond. They are also synthesized in the liver. Their normal value is around 200 to 400 milligram per deciliter. Now, this is all we had for our plasma proteins. So before we begin with the next content, let me first tell you what are the important things you should be knowing in the plasma proteins. Okay, first of all, what are the total proteins? The definition, very important, you should be knowing. You should be knowing all types of plasma proteins that we have, such as albumin, transerythrin, retinol binding protein, and so on. You should be knowing their features, their functions, and the clinical significance we have. Most important content, you should know the definition of acute phase reactant, which proteins will increase during acute phase reactions and which will decrease. Okay, so those were the high yield contents of the last content that we just finished, known as plasma protein. Now the next content that we will continue is porphyrins and hemoglobin. Okay, in order to understand porphyrins, first we need to know what is pyrrole. So let's see what are basically these pyrrole. So pyrrole is an aromatic compound which are made up of 5 rings basically, and they have the formula of C4H4NH. You can see the structure over here. They will have 5 rings, which closed rings. It's an aromatic compound. Porphyrins are formed by these pyrroles. To be more precise, they are formed by joining of 4 pyrrole rings with the help of methynyl bridges, and will have a metal ion linked to the nitrogen atoms of the pyrrole rings. The most important, biologically important porphyrins are existing as a conjugated proteins. They are present usually as a conjugated proteins, and these basically includes hemoglobin, myoglobin, cytochromes, catalase, peroxidase, and tryptophan pyrrolase. So basically, porphyrin will be formed by 4 pyrrole rings which are joined together with the help of methynyl bridges. Okay, now let's see some details about this thing. First one, hemoglobin. It's a type of porphyrin there, which is in a conjugated protein, which is defined as a conjugated protein and made up of 4 subunits. Each of these subunits have a heme group and a polypeptide chain. The heme group of the hemoglobin is basically made up of the porphyrin and the ferrocyanide. Hemoglobin has the ability to bind with oxygen reversibly and therefore allows them to transport oxygen in the body. Myoglobin have a similar structure just like hemoglobin. Only difference is that they are monomer. Myoglobins are found in muscles and they also can reversibly bind with oxygen. Cytochromes. These also contains iron porphyrin which are conjugated with the protein. And their iron porphyrin portion is quite similar to that of the hemoglobin, and they are found basically in the respiratory chain. Also we call it the electron transport chain in the mitochondria. Some of these cytochromes may also perform other functions. For example, microsomal hydroxylation. The fourth one is known as an enzyme. It's called catalase. This also has iron porphyrin and mainly they are found in animals. Their role is to detoxify hydrogen peroxidase. Fifth one we have is called peroxidase. Mainly this is also another enzyme and their role is also to act on hydrogen peroxidase, and they will mainly be found in plants basically. Number 6 are tryptophan pyrrolase. Again an iron porphyrin containing enzyme. They will act on the amino acid tryptophan. Now, this is how a typical pyrrole will look like. Okay, first of all, pyrrole we know is something like this, which will be made up of how many? 5 rings with an NH group here. Their formula was C4H4NH, if you remember. Okay, so 4 of such pyrrole rings will together join to form a porphyrin. And this is how the structure of porphyrin basically looks like. Okay, the parent porphyrin is also known as porphyrin. Okay, you can see here we have 1 pyrrole ring here, 1 pyrrole ring here, 1 pyrrole ring here, 1 over here, 1, 2, 3, 4. They are joined with one another by methynyl bridges. So there's a scientist called Fisher who basically at first proposed the structure of these parent porphyrin called porphyrin. According to his presentation, if you see here, this is how basically our porphyrin will look like with 4 subunits of pyrrole. Carbon atom number 1 to 8 will have different substituents attached to them, and these substituents can be either acetate, propionate, methyl and vinyl. There are 2 types of porphyrins, porphyrin 1 and porphyrin 3. So according to Fisher, the carbon atoms should be attached to different substituents, and there are 2 kinds, porphyrin 1 and porphyrin 3. See here, the substituents are attached symmetrically, that is acetate, propionate, acetate, propionate, acetate, propionate, acetate, propionate. So in a symmetrical manner they are attached. If this is the case, then it is defined as porphyrin 1. So in porphyrin 1, from carbon atom 1 to carbon atom 8, okay, the carbon atoms 1 to carbon atom 8, the substitutes that we have are attached in a symmetrical manner. In porphyrin 3, the manner in which substitutes are added is similar, just like porphyrin 1, except for in the ring 4. So if you see here, AP, 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 but in ring 4, it is no longer symmetric. It's PA, asymmetric. It's becoming asymmetric. So this is a major difference between porphyrin 1 and porphyrin 3. Type 3 porphyrins are the ones that are most commonly found compared to type 1. In human, the important porphyrins are of three kinds: uroporphyrins, coproporphyrins, and protoporphyrins. Okay, keep that in mind. Uroporphyrins are found in the urine. Copro, which refers to fecal material. Coproporphyrins are found in feces. Protoporphyrins are found in blood and tissues. See here, Hem, as we have seen, Hem group of the hemoglobin is the most abundant porphyrin, and it will also have 4 chiral rings. You can see here with the center iron located at the center. Okay, now you can tell me, see here in a symmetrical manner, MV, MV, MP, but here it's becoming asymmetric in number 4 ring. So what do you think? It's a porphyrin 1 or porphyrin 3? So definitely if it's asymmetric, it's your porphyrin 3, our Hem. Okay, now synthesis of porphyrins begins by the help of two things, glycine and succinyl coenzyme. When the succinyl coenzyme gets condensed with glycine, okay, they will mainly synthesize your porphyrin. And for this, we know that succinyl coenzyme is an intermediate of the TCA cycle or citric acid cycle. And this reaction of condensation will be done by the enzyme called ALA synthetase or delta amino levulinic acid. Now, as you can see in this diagram, succinyl coenzyme will get condensed into alpha amino beta-keto adipic acid. And the enzymes that will help will be ALA synthesized, and will also need the presence of glycine. Now, once we got alpha amino beta-keto adipic acid, now this structure will go through a process of decarboxylated to form delta amino levulinic acid. These reactions also will be catalyzed by ALA synthetase, and for this reaction to be carried out, okay, a coenzyme is required, pyridoxal phosphate, PLP. You have learned before from previous lectures what is PLP basically. Now you see here alpha amino beta-keto adipic acid going through a process of decarboxylation, that is removal of carbon dioxide, catalyzed by the enzyme ALA synthetase and coenzyme PLP, and giving us what? Delta amino levulinic acid. Now, two ALA molecules are condensed to form the first pyrrole compound known as porphobilinogen. So that delta amino levulinic acid will now become condensed, two of them, to form the first pyrrole compound called porphobilinogen. And this again is catalyzed by the enzyme called ALA dehydrase. See here, alpha amino levulinic acids getting dehydrated, removal of water by the help of ALA dehydrase to form the first one, porphobilinogen. Different porphyrins can be then formed from this parent porphyrin called porphobilinogen. So the first porphyrin that developed is your porphobilinogen. The exact mechanism by which this synthesis takes place are not basically fully understood. 4 porphobilinogen molecules will react with hydroxymethyl bilane, and this reaction will be catalyzed by the enzyme called uroporphyrinogen 1 synthetase. And as it happens, when the porphobilinogen reacts with hydroxymetabolite, then it can continuously or spontaneously form uroporphyrinogen 1. This is catalyzed by the enzyme called porphyrinogen 3. So basically the whole enzymatic reaction includes two enzymes, uroporphyrinogen 1 synthetase and uroporphyrinogen 3 co-synthetase. Uroporphyrinogens can be then converted into coproporphyrinogens. Later on, coproporphyrinogens can then be converted into protoporphyrinogens. So if you see the diagram, you'll have a better understanding. So 4-hydroxymethyl bilane can spontaneously convert and become uroporphyrinogen. by removing its ammonia. And in the presence of light, uroporphyrin can then become uroporphyrin, or with the help of the enzyme uroporphyrin decarboxylase, uroporphyrin can convert into coproporphyrin 1 or coproporphyrin 1. So the whole reaction also you can see over here, hydroxymethylbenzene, the enzyme uroporphyrin 1 synthetase and uroporphyrin 3 co-synthetase allows it to get converted into uroporphyrin 3, which then later can become uroporphyrin. Then the uroporphyrin 3 can also become coproporphyrin 3 with the help of uroporphyrin decarboxylase. The coproporphyrin 3 can get converted into coproporphyrin 3. The coproporphyrin 3 can also convert into protoporphyrin 3 with the help of the enzyme called coproporphyrin oxidase. The protoporphyrin 3 can again then convert into protoporphyrin 3. Protoporphyrin 3 is the most abundant porphyrin. Among the uroporphyrin, coproporphyrin, this is the most abundant one. It is also the most important porphyrin. Because why? Because heme is synthesized from protoporphyrin 3. Heme group of hemoglobin basically. Heme synthesis is catalyzed by an enzyme called ferrochelatase or heme synthetase. So porphyrin 3 in presence of iron, ferrous iron can ultimately get converted into heme with the help of this enzyme. The heme can then combine with different polypeptides to form different kinds of hemoproteins. Now, this process of regulation, okay, now this porphyrin synthesis is how it is regulated, we need to know. And this regulation is important in order to form heme. So the main enzyme involved in the regulation is our ALA synthetase that develops, if you remember, alpha aminic levanic acid. The regulator is the heme itself. Regulation will occur by the process of repression and depression. Let's see how. So when heme is not being utilized, that is when we have sufficient amount of heme in the body. So if it's not being utilized, the concentration will increase. Now as the concentration increase, it will combine with something known as a aporepressor and form something called repressor. These repressor will act on the enzyme that is involved in the regulation called ALA synthetase, and thereby will prevent the synthesis of ALA synthetase. This, if we don't have sufficient amount of ALA synthetase, this will lead to decrease of synthesis of porphyrins. Similarly, when heme is utilized in the body, when the concentration is decreased, the synthesis of ALA synthetase is increased. This will further increase the synthesis of porphyrin. So that's how basically the porphyrin synthesis is regulated. Okay, so we'll stop here today and we'll further continue in the next class with our blood biochemistry here. So for this time, you first must know this whole process. Okay, you should be knowing what is a pyrrole ring, how a porphyrin is, the structure of porphyrin is made up of, how the synthesis of porphyrin takes place, types of porphyrin such as uroporphyrin, coproporphyrin, proteoporphyrin, and how this porphyrin synthesis is regulated. It's very important content. Please do remember how the porphyrin is synthesized, the whole mechanism. You must remember, including the enzymes that is involved. So that's all for today. Thank you.